Can you see the heading with seed banks? Hello?
Yes.
Okay. So with seed banks is where seeds are stored. So the question is, where are the weed seeds stored after formation and shedding? The answer is they are stored in the soil, and we have soil seed bank. They may remain on the mother plant, what we refer to as the aerial seed bank. So what exactly is the seed bank? After weed seed shedding, seeds are deposited on the soil surface. The weed seeds could be incorporated in the soil during tillage operations. So, what constitutes the weed seed bank are the seeds on the soil surface plus seeds in the soil. They will give us the soil seed bank. So, seeds may remain attached on the plant that is known as the aerial seed bank, but eventually they will come into the soil, probably through the plowing operation. So here we've got weed seeds which are on the soil surface, and we've got weed seedling emergence. This is withdrawal of the weed seeds from the soil. This could be an investment route because some of the plants will flower and produce more weed seeds, which are channeled back into the soil. Here we've got shamba grass, which has got cylinder shaped seeds, which are on the ground. These are now part of the weed seed bank. If I can show you the soil profile, probably O up to A may constitute the soil profile which is referred as the seed bank. Below this, which could be B, probably there are no weed seeds. Uh, I just want to use this diagram to illustrate what is happening to the weed seed bank. The weed seed bank is represented by that box in the center, if you may call it. We have got the seeds which could be in an innate dormancy state, induced dormancy, or enforced dormancy. They have to exist in either one of those forms. Now, let us look at processes which can withdraw seeds from the seed bank. This route, physiological death, at the end of seed longevity, seeds may die. We can use implements which can damage weed seeds like plows, harrows. The seeds may actually fail. They may fail to germinate, or they may germinate, then they are attacked by pathogens, and then they die. Or they could be eaten by animals. This is a predation. The other route is through germination, which result in a plant, and this plant will flower and form seeds and add seeds into the soil through the route, what we call seed rain. Sometimes we talk of seed rain, addition of weed seeds from the plants. The other route is that of weed seeds from dispersal. And we have been discussing dispersal of weed seeds. So weeds can be dispersed and come into the seed bank. So this is exactly what we want to talk about. Probably this is a clear model of weed seed dynamics. Now let us look at processes within the seed bank. We have got non-dormant seed bank or non-dormant seeds which are ready to germinate. We also have dormant weed seeds which are not ready to germinate. So there's a sort of an equilibrium where weed seeds can change from being dormant to be non-dormant. And we now know various forms of dormancy which can occur. Again, we can look at the weed seed losses. These are the white roots, these big white arrows are showing withdrawal. So we can have a predation by birds, mice, and insects. We can also have a decay. Pathogens and fungi, they can actually reduce weed seed bank. Then some may germinate and die. It could be through natural death or through weed control measures. Others may escape weed control measures. They will germinate, live, and produce seeds, what we refer to as weed seed rain. Or this may be referred to as a FDI, foreign direct investment of seeds. And we may use this monetary language, to describe what is happening. Seed dispersal, which involves rain, wind, animals, and machinery, can bring in weed seeds into the weed seed bank. So we have this weed seed bank dynamics. It's like a normal bank where you have deposits and withdrawals, depending on what is happening in the bank. Some withdrawals may involve investment, and then the seeds or money is deposited back into the bank, something like that. Or this may come as a foreign, foreign direct investment from other areas, and you get these seeds. So this is the model. In fact, when we control weeds, we are actually influencing this weed seed bank dynamics. Probably we need to explain further what is a seed, what is a weed seed bank. Probably it's the reserves of viable weed seeds present on the soil surface and in the soil. It can consist of new seeds shed by the plant or older seeds in the soil for several years. Seed bank is an indication of the past and present weed populations. There are enormous numbers of weed seeds in the soil. Some weed seeds die within a few years, but others persist for a long time. Now, the density of weed seeds are linked to the cropping history of the land. Seed density is influenced by the past farming and may vary from field to field. Thompson and Grime actually grouped seed banks into two parts. The first group they referred to as the transient seeds. This is where you have grasses which do not persist for longer than 1 year. Then you have the second group, which are known as persistent seed banks, where you have species of a wide range of habitats. In grasslands, the majority of the seed bank is located near the soil surface, and nearly the entire seed population is located in the 15 centimeters of soil. CA, which is conservation agriculture systems, we have seed banks which are closer to the soil surface. Seeds are in a better position to germinate and interfere with crop production. That is in the CA. Under good weed management, the seed bank could be more easily reduced. But I don't know whether we can actually reduce weed seed banks to a level where we are not going to worry about weed management. Now let us look at the tillage effects. Now, tillage affects the vertical distribution of seeds in the soil. We have been talking about weed dispersal within short distances within the soil, what we call vertical seed dispersal. In undisturbed conditions, the weed seeds remain near the soil surface, like in conservation agriculture. Tillage with the moldboard plow buries the weed seeds deeper. In reduced tillage, the seeds are close to the soil surface. So that is the difference. Where we have conventional tillage, there could be more or less uniform distribution within the soil profile. But in reduced tillage, weed seeds are concentrated towards the soil surface. No wonder why the reduced tillage or conservation agriculture could be associated with the high density of of weeds after germination. Now, if weed seeds are closer to the soil surface, this may reduce dormancy and encourage germination. Probably that explains why we may have a lot of weeds in reduced tillage. Here I've got a diagram showing three different tillage systems. We have got the use of the mold body plow and what is known as the chisel plow and no-till plow and no-till, sorry. Now, if we look at the distribution of weed seeds in the soil profile, starting from 0 going up to 6 inches, according to this paper. Now, there is more or less uniform distribution, or there is less weed seeds near the soil surface, and there are more at the bottom. With the chisel plow, which is of less soil disturbance, we may have this kind of distribution where you have more seeds at the center and at the top, less seeds at the bottom, or we have got more of the weed seeds near the surface and they decrease as you go down. This is quite unique in the no-tillage system where the distribution of weed seeds are very close to the surface. As you go down, there are very few seeds, as shown by these two diagrams. This could have been assembled at various, probably there were 6 sites where this was done. Now there's a study, maybe I will have to give you this paper by Mavunganidze et al. In 2009, who studied the weed seed banks in Mzarabani area. They reported that the 0 to 10 centimeters had higher weed seed numbers than the 10 to 20 and the 20 to 30 centimeter soil depth levels. Probably this is a reflection of the tillage systems which were being done. Another point they reported is that fields which had been cultivated for a long time, let's say more than 20 years, had larger seed banks than recently opened fields. So most of these fields which have been under cultivation for a long time have a lot of weed seeds. So the soil acts as a medium of seed storage and the growth of weedy plants. Now, what we are going to do is to examine how weed seeds can be lost from the weed seed bank, and this can possibly affect the weed seed bank size. These are the withdrawal routes. So loss of seed from the soil. Seeds are eaten by rodents and insects. They may decay due to microbes. They may germinate and emerge. Probably the plant may die. One of the experiments done by Biru, it's known as the Biru experiments, indicated that there are some weed seeds which can stay being alive in the soil for 120 years and it would still be viable. In the Duvel experiments, some may stay for 39 years and they would still be viable. But of course, these seeds may decay, or they may germinate, or they may be eaten by rodents. Now, seed longevity can increase with all species as the depth of burial is increased. It tends to store these seeds. So seed longevity depends on depth of seed burial, amount and intensity of soil cultivation, crop management, and weed control programs uh, have got a big impact on the changes in the seed bank. When we look at crop management, there are several processes which I will show you later on. Soil seed bank can be reduced by applying herbicides. It's part of crop management. For example, withdrawal of herbicides leads to increase of weed seed bank to its original size within 1 year. You may find that the experience of the newly resettled farmers in the commercial farms was that the previous farmers were using herbicides, but when the new farmers entered in, they did not apply herbicides. So they faced very serious weed problems to the extent that some crops were actually abandoned. But probably as time went on, people started to use some of these herbicides. What about entry of seeds into the soil? Now, source of seeds, it could be weeds that are allowed to mature to produce a seed, then they just add these seeds into the soil. So we have weed seed production of standing weeds. We have got seeds that can also enter a field from outside sources. This could be through dispersal. So the primary seed dispersal methods are wind, water, animals, and humans, as we have discussed. Surface irrigation water is a potential to distribute weed seeds as well. Now, crop seeds could be contaminated by weed seeds. For example, wheat seed can be contaminated by Datura stramonium seeds. In fact, if wheat is delivered to the GMB, what inspectors they look at is the Datura stramonium seeds. If wheat is contaminated, they reject the wheat consignment until that wheat has been cleaned. You know, Datura stramonium seeds are toxic, they are poisonous. Sorghum, pearl millet, and finger millet seeds are also likely to be contaminated by weed seeds. Equipment movement can distribute weed seeds. So many weed seeds pass through the digestive tracts of animals and remain viable. So when we apply manure, we could also be adding weed seeds to the seed bank. Wildlife also spread these seeds. Let us look at the Germination of weed seeds and emergence. Seeds can either germinate or remain dormant. And we now know the three seed dormancy types: innate dormancy, induced dormancy, and enforced dormancy. Where innate dormancy is the inherited characteristics, where we look at embryo status and the resistance of seed coverings to water and gas exchange. Induced dormancy is germination does not occur even when the conditions are not, even the conditions are favorable, because the seeds have a memory of which is stored inside them, which can tell them not to germinate, and this process is not reversible. So first, if you put seeds in an unfavorable condition, they are not going to germinate. But when you reverse these conditions, the seeds remain dormant, as if the conditions are not favorable, because the information inside the seed needs time to change so that dormancy can be broken. But enforced dormancy, it can be caused by environmental conditions such as low temperature or lack of moisture, oxygen, and light. But this can be reversible. That is the difference. Now, let us look at the effect of depth of seed barrier. This can affect the germination and emergence. The ability of seeds to emerge from various depths depends on the soil and the seed weight, or probably seed size. For example, large seeds are able to emerge from greater depths. Light penetration in the soil, for example, silt, clay, loam, and sandy soil, is about 22 millimeters. So it is this light which can determine whether seeds are going to germinate or not. Now, what are the factors that affect seed bank size? Here we are looking at the total number of seeds within the soil. Seed bank, that is the seed bank size. It is determined by the number of factors, including the previous history of the land. That could be weed control, weed species composition, weed population, tillage depth, and frequency of tillage, dormancy characteristics of weed seeds, and the lifespan, which is longevity of the weed seeds. But probably this statement we may need to simplify it or clarify a few things. We have also management of the seed bank. There are ways of reducing the input of seeds into the seed bank, which could be use of herbicides, crop rotations, tillage, mulching, reduce the weed seed dispersal. Probably that information could be summarized in this diagram. Now, on the left, we have got crop management practices, where we have primary tillage, mulch, fallow period, inorganic fertilizers, cattle manure, secondary tillage, harrows, ox cultivators, ox plow. When we are doing weed control during secondary tillage, we may also have crop rotation, intercropping, crop spacing, and use of herbicides. So what happens with the crop management, depending on the efficiency, it can affect this root seed deposits in the soil seed in the soil seed bank, which is this portion here at the end here. Or these activities can also have an impact on seed withdrawals from the seed bank. Right. The other issue is that as these processes are taking place, they interact with the agroecology. Soil type, rainfall, and the temperature may interact with these processes and affect with the seed withdrawals, the red root, or with the seed deposit, the blue root. Then we can also have a seed dispersal influencing this seed bank. Inside there, we can have insects and fungi, birds also acting on weed seeds. We also have a seed germination process, which can result in other plants also producing weed seeds. There can be dormancy, which can also influence the seed longevity. So all these processes can affect the weed seed bank size. So the key drivers of weed density and diversity could be the crop management. And the crop management can actually interact with agroecology, soil type, rainfall, temperature to influence what is present in the soil. So this can summarize what is happening to the weed seed bank. We want to look at the methods which are used to determine soil seed banks. One of the most common methods is known as the weed seed germination method. What you do, you use augers, preferably 5 centimeters in diameter, and you take soil samples from 0 to 10, 10 to 20, and 20 to 30, so that you can see the weed distribution. Probably about 100 sampling points are required. However, increasing sampling will increase precision. So if you increase your sampling size, you also increase precision. Now, the soil cores from the same depth are bulked together and passed through a 2 millimeter sieve. These soils are then spread in 55 centimeter times 30 centimeter asbestos trays. Last year, we had the students who had to use plastic polythene bags to do this study instead of these asbestos trays. So the trays are watered as necessary. Weeds that emerge are identified, counted, pulled, and discarded. The soil is disturbed after 1 month to encourage germination. The duration could be more than 3 months so that you can exhaust the weed seed germination. This is the method which was used by Mavunganidze et al. In 2009 to determine weed seed banks in Mzarabani area in Zimbabwe. So this method could be used to study weed seed banks. We also have the direct seed extraction method. I must say this one is a bit difficult because you need to recognize and identify weed seeds from the soil particles, which is quite cumbersome. Weed seeds have to be separated from the soil using flotation and sieving methods. Soil samples could be soaked in a solution of sodium hexametaphosphate, or you can just use the sodium bicarbonate for a short time. I remember at Henderson Research Station, we used sodium bicarbonate, and even at Tobacco Research Board, a student there used the sodium bicarbonate to extract Striga asiatica seeds and Striga gesneroides seeds from the soil. A jet of water is used to remove clay and silt particles. Once seeds have been isolated, they can be identified. Viability tests will be done using the tetrasorium test so that you can distinguish the dead seeds and the live seeds. So it's quite a cumbersome process. So the first one, where you germinate seeds, this one, the weed seed germination method, is I think it's better than the direct seed extraction method. But anyway, people have different preferences. Now, there could be a correlation between the soil seed bank and the weed populations in the soil. So there's a paper which can explain this. Probably I will have to send it to you, or you may have to look for it if you wanted to explore more about weed seed banks. So there's a linear relationship between weeds in the soil, weed seeds in the soil, and the weeds that emerge. So all these graphs are pointing to that effect. If you read that paper, you will get explanation of this. And also, this is the stuff from that paper showing linear regressions or showing that there's a relationship between weed seeds in the soil and those weed seedlings that emerge. So these are some of the references which you may have to read concerning weed seed banks. So we have got several reference materials which you may have a look at. So I'll end here. Then you can ask me some questions